Hi class, Dr. Jim here. Now this is the second video that we have accompanying the monohybrid dihybrid crosses. Now this one I said make sure you do after you get the monohybrid, and this is when we talk about special situations. There's still monohybrid crosses that we're talking about, so again 4 boxes, but in this situation we have a little bit different things going on. We either have codominance where genes get shared, or we have incomplete dominance where you see a mixing of the two traits, or you have X-linked genes. And so again, all these require some different little nuances that you have to do. And so what I really want you to do is make sure that you understand the monohybrid cross first before you really attempt these problems. Because if you don't understand the monohybrid, this is going to make it even more confusing to you. So make sure that if you're watching this video, you can watch it to kind of get an idea of what we're going to be doing. But make sure that until you have a good understanding of the monohybrid, that you don't do these problems also. Okay, until you get a good understanding of those things. So let's go and see what these special situations are going to be. All right, so the first one is looking at how to perform a Punnett square with a codominant trait. And codominant means that you actually have 2 alleles or more than 2 alleles for one gene, or basically you have multiple traits for one gene. Okay, and so the typical one we use in class is the ABO blood type, and so you can have A type blood, B type blood, AB type blood, or O. And so we'll look at a situation like that and how to figure one of those crosses out and be successful at that. The next one we're going to be looking at is how to do an incomplete dominant problem. And an incomplete dominant represents where basically you have the dominant and recessive traits like you've seen before, like tall versus short, but the heterozygous condition is kind of a mixture of the two. So the typical one that's always used in class is talking about the snapdragon flower, where you have the red snapdragon flower and the white snapdragon flower. The red is dominant, the white is recessive. But when you put the heterozygous, when you have one red and one white, instead of getting red, you actually get pink, where the two mix together, and that's called incomplete dominance. You can kind of think of it too tall and short, and in the middle you get medium height. Okay, and then the last one deals with the X chromosome, and so this one is associated with genes that are found on the X chromosome. It doesn't mean these are all sex-related type of things or determining male versus female, but what we look at is genes that are actually found on the X chromosome. And so the disadvantage comes to males because when we have, in our chromosomes, we only have one X and one Y, and you're going to see the Y is much, much shorter than the X, and that leads to problems for genes that are not found on the Y. So we only have one copy of the gene. And where this can be an issue is things like hemophilia or colorblindness, and that's why you typically tend to see more males have these things, because we're unlucky to only have one copy of the gene. So if we have a bad copy, that's what we're stuck with. We don't have a backup one like the females do where they have 2 copies. Okay? So we're going to look at both of these situations, or all 3 of these situations, and kind of see how to solve problems for all these types of things. Okay? So the first one we're going to look at is codominance, and this is where some traits are actually controlled by multiple alleles. And so that means that basically you have a gene that exists in several allelic forms. You might have 6 different alleles, but you only get to pick 2 of those 6. Or you might have 8, and again, you can only pick 2, or 10, and you can only pick 2 of those 10. Okay, and that's what this is. And so the one that we normally refer to in class is the ABO blood type, and that is basically where you could have A-type blood, you could have B-type blood, you could have AB, or you have O. And I'll talk a little bit about how that comes about with the alleles and how we figure that out. Now, the alleles for the ABO is simple. Basically, you either have the A allele, you have the B allele if you have the B blood, or you have the little I allele if you have O. Okay, and so really you have 2 different genes or 2 different alleles in this situation that both are dominant. And in this situation, and then the little I, the O, you have the recessive. And so 2 dominant and 1 recessive in this case. And so we call this the codominance because more than one allele is fully expressed. And so where the situation gets really interesting is if you have both the A and the B, you actually have AB blood. You don't have just A, or you just don't have B. You actually have AB. It's where the dominance is shared between the 2. So we'll see a problem. We'll do one and see how it works out for you. Okay. So how does this work? So if you have the phenotype, if you have the A-type blood, you're going to have the genotype that's either big A, or big A, O. Okay. If you have the B type blood, you're going to have the big B, or the big B or O type blood. Okay. If you have AB, you can only have A and you can only have B put together. And if you have O, you're going to have the little I and little I. Okay, and so that represents the different genotypes. So where it gets interesting is in this situation, and this is how you can explain where if you have both an A plus parent and a B plus parent, and you get an O child out, how did that work? Okay, well, it means that both parents are actually carrying the O allele in their genes, but they were expressing the A or the B because the A and B are both dominant. They hide the recessive trait. Okay, and we'll look at a problem like that later on. All right. So let's do a problem and see if this makes any sense. Again, this is monohybrid, so we're only talking about four offspring, and we're looking at just one gene. So let's do the problem and see if you can do this. So if an AB-type man marries an O-type woman, what is the percent chance that the children will have A-type blood, B-type blood, or O-type blood? So if you want to try this without going through this or having me go through it, pause now, then I'll stop, and then if you want to see me how to solve this, you can watch it and then go back and try and do it yourself. So let's have you guys pause. If you want to pause now. And then if you didn't pause or you're continuing on, here's what we're going to actually do. I'll show you how to solve it. So the first thing you want to do is figure out what are the genotypes. We know the phenotypes because we know the man has AB blood and the woman has O. So what are going to be the genotypes? Well, the man's pretty easy because you know the only genotype he can have is the big I with the A and the big I with the B because he has AB type blood. Okay, so his will go on the side. And then the woman who is O type blood is going to be pretty easy too because it'll be little I, little I. Okay, and that's what her genotype is going to be, and we put that on the top. Now you just do it like a normal monohybrid would. Okay, and you put it in, you have the big A with the little I, the big A with the little I, and then the big B with the little I and the big B with the little I. Okay, simple, just like the monohybrids. Okay, now we want to figure out what are we looking for. So they're asking for what is the percent chance. So again, you count how many of the children are going to have the A blood, how many are going to have the B blood. We can see here this child has an A and this child has an A, so they're going to have A blood, and those guys will have 50, or it'll be 2 out of the 4 possibilities, so 50%. Same thing with the B-type blood. We have 1, 2. 2 out of 4 is 50%. Okay. The last one is O-type blood, and the only way you get O is if you have the genotype little I, or as in the case of the mom, little I, little I. And in any of these situations, do you see little I, little I? No. So you don't see any of those, so the percentage would be 0%. And we can figure them out right here. A-type blood is 2 out of 4, that's 50%. B-type blood is 2 out of 4 or 50%, and then the O-type blood is 0 out of 4 or 0%. And that's how you answer the question. So again, I have a couple of these in the practice problems on Blackboard. Make sure you practice, take a look at these things, and kind of get a feel of how to do these. These are also in the lab sheets that we have on the handouts this week as well. So practice these problems. Okay. So now the next thing that we're going to talk about is incomplete dominance. And incomplete is where you have, again, a mixing of the two traits. So we're talking about just 2 alleles, either red or white, or maybe tall and short. But when you get the heterozygous, instead of getting just the tall phenotype, you actually get a mix of the two, or blending of the two, and you get either medium height or pink in the case of red or white. Now again, the situation that we talk about here is typically the snapdragon plant, and again, homozygous red would have the red phenotype, the homozygous white would have the white, but the heterozygote, which is the intermediate, is going to have the pink instead of having the normal red, which you'd see in the monohybrid. The phenotype reveals the genotype without a test cross, so you can kind of look just based on what they look like, okay? And that's the easy one because you know if they're pink, they're going to be heterozygous. If they're one color red, they're going to be dominant. If they're white, they're recessive. So really, incomplete dominance is the easiest of all the monohybrid crosses that you could do, okay? So if you look, these 2 are pink. We know that these 2 are pink, so they know that they're in the heterozygous condition. And so when we put these together, you can see that when they're red, they're homozygous dominant or have the 2 big R's with the 1s. The pink are the heterozygous, so they have the 1 and the 2. And then the white is going to be both of the recessive or the 2 in this case. They use the numbers here. You could use the big letters and small letters. I use the big letters, small letters, but this is taken from your book, and so you can do it either way in this thing. It's where the 2 get mixed together. Okay. Now, we actually do have human traits that do this, and so I just bring this up. We're not going to do a problem with this or anything, but I just want you to be aware there's one called hypercholesterolemia, which is called FH, or familial hypercholesterolemia, where actually homozygotes for the mutant allele develop fatty deposits in the skin and tendons and have heart attacks when they're really young. So the homozygotes that are for this trait, the FH minus or the recessive traits, are going to have fatty deposits. Homozygotes have a normal allele, don't have this disorder. Most of us are this. And then heterozygotes might suffer from heart attacks in early adulthood without it. And so typically these people will come into the hospital and they could be skinny as a rail and still have this really abnormal cholesterol levels. And you know, the ones that have the double mutant are going to have really, really high cholesterol. And you're like, you know, they eat healthy, they look skinny, everything else, and they have this extremely high cholesterol. And so this is just a human example of showing you where this incomplete dominance is. I'm not going to use it as a problem or anything, but I just want you to be aware of it. Okay? Now the last situation that we're going to talk about is the X-linked inheritance. And this is when we talk about the X and the Y. Okay, it's not talking about are you a female or a male, but what you're looking at is the genes that are found on the X chromosome. Okay? All mammals have at least one X. If you're female, you're lucky and you have two X's. If you're a male, you only have one X and the other one's Y. You can kind of see the X is nice and big, the Y is a little shrimpy guy here. And these are called the sex chromosomes because this determines what your sex is. Are you male or you're female? If you're male, you're XY. If you're female, you're XX. Okay? Just kind of shows you what the differences are. So you can see that this is a short little stubby guy, and the genes that are above this level here are only found on this X chromosome. They're not found on the Y. And that's really important because in males, if you have a defect in one of these genes, you do not have a backup on the Y to help fix that defect or help cover that defect. And so that's where the problem comes. And we're going to look at some of these disorders where we worry about this X-linked inheritance. So the term X-linked inheritance is used for genes that have nothing to do with gender. Okay, it's just because they're found on the X chromosome. It doesn't mean male or female in this case. Okay, X-linked genes are carried on the X chromosome, and these were first discovered in the early 1900s on the fruit flies. And fruit flies have a very similar chromosome pattern that we do in having X's and Y's to determine sex. And so this is where they actually figured out the whole X-linked traits. And so one of the things that in fruit flies is that you can have white eyes versus red eyes. And typically fruit flies have red eyes. Okay? But X, or the white eye gene, is actually found on the X. And so if you have a defective X, you actually get white eyes. And so if you put this through and you look at the X's, the X always comes from mom for the boys. And even in the females, you have the reciprocal, you have one good X, and that's all you need is one good X and then one little r. Okay? And as you put it through the second generation, so you mate these guys to the second generation, you see this pop up. And we can figure these out based on heredity and things like this. And so you see these throughout the family histories. And probably the most famous one that we've seen in human heredity is dealing with hemophilia and the czars of Russia and things like this, and the royalty of the European times. And so we'll look at a couple of these different situations that are called X-linked inheritance. And so several X-linked recessive disorders occur in humans, one of them being colorblindness. Okay, it's for blue sensitive protein is autosomal, and red and green are on the X. Okay, muscular dystrophy, or MDS. So you probably have heard of MDS before. Maybe you've seen the telethon on Labor Day. And so MDS is actually carried on the X chromosome. So if you have a bad copy of that gene on the X, males tend to suffer more than females because they only have one copy of the X. But it does happen in females. Same with hemophilia. Hemophilia is a situation where people tend to bleed because they're lacking the factor 8 or factor 9 protein that helps clot the blood. And so again, these are factors that are found on the X chromosome. And so if you have the bad copy that don't make the correct protein, you have this disorder. Okay? So let's try a couple problems with these things. So the first one we'll do is colorblindness. And so this is an X-linked trait, and they'll tell you in the problem that it's an X-linked trait. And if it's normal vision man marries a woman who is colorblind, what is the % chance to have a colorblind son? How about a colorblind daughter? So we have to look at the X's and Y's. So this is where now it gets a little tricky. This is the only trick is that you have to remember sons versus daughters, because now we're talking about X's, XX, and XY when you put these things together. So that's important, okay? So let's figure out the genotype. So a male genotype for colorblindness, if he's normal, is going to be XY because he has a normal X and he doesn't have that gene on the Y, so he's XY. In the female, and so we put that down here, in the female, she is colorblind. So we show this as a little c over off to the side. So she has both X's because she's female, but in her situation, in order to be colorblind, both of the X's have to be mutated to show the colorblindness, okay? So we designate this with the little c on the side. So we put her on top, and we just figure out the monohybrid, just as before. So we see here, here's the X with the C and the X, and the X with the C and the X. And so you have one good gene, this X, and one bad that has the C, okay? In the males, all you get is the X from the mom. So you have the X with the C here and the X from the C here. So now we want to figure out how many are going to be colorblind sons and how many are going to be colorblind daughters. And so what you have to do is make sure that you look at the X's, XX's, and XY's to determine the sex of the child, okay? So for the males, all we're concerned with is these bottom boxes down here because all we're concerned with is the XY, okay? So these are the boys, XY. You can't have boys with XX, so these are the two you look like. And so what is the percent chance to be colorblind? Anytime you see the C represents colorblindness. And so in males, since you only have one copy of the X, if you have the C, you're going to be colorblind. So we have 1, 2 colorblind sons. So that would be a 100% chance that they're going to be colorblind. How about for the female? Well, in the female, in this situation, they have one good gene, which is this X, and one bad gene. As long as you have one good gene, you're not going to be colorblind. So in this situation, again, you have two females, okay? And both of them are going to have normal vision. So they're 100% or 0% that they're going to be colorblind in this case. And so you can figure this out. So if you have a colorblind son, it's going to be 2 out of 2, which is 100%. Okay, you see that? Because again, both copies of their X's have the colorblind gene. Okay, whereas in the females, they have one colorblind gene X for both, but one good. And when you have one good, that blocks out the bad. Okay, and so in this situation, since the colorblind daughter would be XC, XC, we don't have any of those, and so it's 0 out of 2 or 0%. And so hopefully you can figure this out. Let's try hemophilia, and I'll let you guys do this problem, and I'll show you how to work this out. I'll read the problem, you can pause it, and then do it. It works just like the colorblindness does with the X, but use the H instead of C for colorblindness, use the H for hemophilia. So let's figure this one out. Hemophilia is another X-linked trait. A hemophilic man marries a normal woman whose father was hemophilic. What are the chances their son will be a hemophilic? What about their daughter? So pause here, and then we'll come back and answer the question. So pause now. Okay, if you paused and you were able to answer the question, good for you. If you weren't and you want to work through it, let's work through it. And let's check your answer to see if you were right. Okay, so let's figure out what is the male's phenotype, or the genotype. We know he's a hemophiliac, so he has the one bad gene, okay? He's XY, but that X has the H, or the hemophiliac gene on it, okay? Because he's a hemophiliac. Now for the woman, we see that she's normal, so we know she at least has one copy, good copy of the X. Now the other X comes from their dad, and we see that her dad was a hemophiliac, so she got a bad copy from dad. So her genotype is going to be X with an XH because she has a hemophiliac dad. And so she got one bad copy, but again, she's normal because she has one good copy over here as well. Okay? So hers goes up here. Let's put them together. So we look at the females. This X drops down and this XH comes across, and we can see that we have an X with an H and an X, a normal one, and we have an X with an H and an X with an H. Okay, here. Here we have the son, XY, and an X and an H and a Y. Okay, so we can see in this situation, one of the boys is normal and the other one's going to have hemophilia. Likewise, in this situation, one of the girls are going to be normal because she has a good gene here, but in this situation, both X's have the hemophilia gene, so she's going to have it. So in this case, we say 1 out of 2, or 50% are going to be hemophiliac for women, and 1 out of 2 are going to be hemophiliac for boys. Okay, so here you go. 1 out of 2 is 50%, and for the daughter, 1 out of 2 is 50%. Hopefully you're able to do that. Again, practice some of these problems. You'll see more of these problems on Blackboard and other things so you can practice, but give it a try and see if you can do it. Okay. Okay, so we summarized, we looked at codominance. This is when you have multiple alleles. You might have 8 alleles, but you can only have 2 in the case of the ABO blood type. So when you think of ABO, think of the codominance. Incomplete is when you share. So you have the dominant, which would be one color, the recessive, which is another, and then the heterozygous condition, you have actually a blending of the 2 colors. And then finally, the X-linked is when the genes are found in the X. And again, you have to pay attention to the male and female with the XY and the XX, and you got to see where these genes are. Males are at a disadvantage because they only get 1 copy of X, and if they get the bad copy from mom, that's what they're stuck with. Okay? With that, we've come to the end of the lecture. If you have any questions, please feel free to email me, send me, messages, or ask me in class. And again, progress on to the dihybrids once you've figured this one out. And I'll talk to you next time. Thanks for watching.